I'm Alison Cranage, and I'm the moderator of this full scientific session using genomics to track and defeat COVID-19, cholera and malaria. Thank you for joining us. This presentation is one of 3 pre-recorded videos focusing on a speaker's material before all the panelists come together in February during the live discussion session. In this video, Dr. Abdoulaye Djimde will be discussing genomic surveillance of malaria. Midway through the video, Dr. Estee Turuk will be joining Djimde to delve further into the material.
Greetings, ladies and gentlemen. My name is Abdoulaye Djimde from the University of Science and Technologies of Bamako in Mali. I will be discussing about how we've been trying to use genomics to help in the elimination of malaria in sub-Saharan Africa. As many of you may know, malaria is still a very important problem. Kills nearly 400,000 people a year, more than 90% of which are in sub-Saharan Africa. So it's a major problem for us. And this graph shows the trend that the disease has been taking in the blue open circles. This is the actual trends of the disease in the last few years. As you can see, there was a downward trend and then a sort of stalling of this decrease in the last few years. If everything went according to plan, we would have been with the open green circles and with a steady decrease of the disease nearing zero around 2030. That's the global technical strategy that the World Health Organization estimated. And as you can see, we are completely off track of that of those goals. So in order to eliminate malaria, we need to do more, we need to do better, and we need to do things differently in order to get us back on track. That's where a group of African geneticists came together a few years ago to form a network that we initially called Plasmodium Diversity Network Africa, and it evolved into Pathogens Genomic Diversity Network Africa, a network including scientists from 16 different sub-Saharan African countries, including from East to Central to West Africa, Anglophone, Lusophone, and Francophone African countries. Diversity in terms of malaria epidemiology and malaria drug resistance and malaria transmission. This group has been conducting genetic studies. In one of our latest studies has been trying to learn from the genetic diversity of the malaria parasite. We sampled parasites and collected genetic information from 15 countries in sub-Saharan Africa. We sequenced more than 2,000 Plasmodium falciparum genomes, Plasmodium falciparum being the main malaria parasite in sub-Saharan Africa. We did both molecular studies as well as complex bioinformatics analysis in order to try to understand the genetic diversity of the malaria parasite and how we can derive knowledge from that genetic diversity in order to help advise the national malaria control programs that are responsible for making policies in the fight against malaria. This graph shows that there are 6 different subpopulations of parasites. The circles that show the different groupings of parasites that we are able to identify by doing this study. I should say that before this work, we used to think that the parasite, in sub-Saharan Africa were so diverse that it was not possible to group them together. This work that was focused exclusively on African parasites demonstrate that there actually are discrete subpopulations of parasites that have genetically sufficiently different that you can group them together, and they actually do group in according to the major geographical regions. As you can see, the big circle represents West Africa. We have East Africans grouping together, the Central African parasites grouping together, and Southern African parasites also grouping together. And we have Ethiopian parasite from Ethiopia that actually form a separate group from the others. So what one can try to learn from this is, since we clearly see that there are genetic differences in these parasite populations, they may react differently when you try to apply interventions. Would it be drug interventions or vaccine interventions or insecticide interventions on those parasites? So the one-size-fits-all type of policy, where you have a recommendation that is blanketly made and need to be applied everywhere, is probably not the best solution in attacking malaria in sub-Saharan Africa. The same study shows on this graph here what we call signatures of selection. It actually detects decreased diversity in the malaria parasite genetics. And we were able to show that the expected decreased diversities that were already known in around the genes responsible for resistance to the main antimalarial drugs, chloroquine, sulfadoxine, and pyrimethamine, were again demonstrated in this work, being sort of a sort of validation of the approach. We see a new signature of selection on chromosome 12 that indicates that there may be a new event, genetic event that are likely to be the beginning of the emergence of resistance to the main antimalarial drugs that are being used, the artemisinin-based combination therapies. And this is more noticeable in Malawi and in Ghana. So this is information and early warning about the possible development of antimalarial resistance in Malawi and in Ghana. And we are going to continue this work to further characterize this early warning. The same work demonstrated that there is flow of genetic material of the parasites across the continent. It appears that the malaria parasite, may have originated from the central of Africa in the Democratic Republic of Congo and spread in different directions towards the east, the west, and towards southern Africa. And so now we see that although this parasite population may have come from the same pool in DRC, they have now discrete subpopulations that are evident from the genetic work that I just presented. So this technical work, for them to actually bear fruit and be used, the scientists need actually to go beyond their laboratories and engage with policymakers and ensure that the national malaria control programs are well educated and well informed about this type of work and the potential of this type of work, so that they would be ready to take it up, to take the results up, and to use them in conjunction with other data for disease policy making. In conclusion, we think that this shows an effective use of genomic to inform policy that will require much more than building facilities and training scientists. We need to undertake early dialogue and engaging with the stakeholders to ensure success. I thank you for your attention.
Thank you, Djimde. Now, Estee will join the conversation. Welcome.
Okay, well, thank you very much, Djimde, for a really, really interesting talk. First of all, I'd like to ask you a little bit more about the Pathogen Genomic Diversity Network that you mentioned. Whose idea was it? How did it start? How did it grow? Can you give us some insights into that?
Yeah, this is actually an interesting question. It started at Kingston in Cambridge. A few of us were all the initial members are all Dominic Kwiatkowski's former students, and we were all part of the malaria gen community. And those days, every year there was a GEM meeting, Genomic Epidemiology of Malaria meeting that Dominic convened at Kingston. And all of them, we regularly attend that meeting. And we realized that the African parasites were always presented, the genetics of African parasites were always presented in the context of Asian parasites and Latin American parasites. They were just used like negative control for all of the work that was going on. And we were a bit frustrated by that. We said, well, there might be more into African parasites than just being negative controls. So we need to get together and actually focus on that and study African parasite for their own sake. That's how it started. We were just 4 or 5 of us. And over a coffee break, we discussed the issue, and we decided to come together and to do something about it. And then we had no money. We just had the idea. And we approached Dominic and explained to him what we wanted to do. And he was generous enough to, he liked the idea and to get us together at Oxford. For a 2-day brainstorming meeting so that we can sort of put some more thought on this idea. And that's how it started. He funded that one meeting, and he funded another meeting in Ghana where we had, from 5, we went to 7 people. Mm-hmm. And from there, we really designed the concept, and we looked around. We said, well, if we want to capture diversity, then we need to include the diversity, both the language diversity, the geopolitical diversity, and the epidemiological diversity. So we invited colleagues that we know could represent the missing pieces in our puzzle, and that's how the pDNA initially was Plasmodium DNA, and then it recently evolved in pathogens DNA, because we are now expanding beyond malaria to include things like COVID or AMR, etc.
Yes, in fact, I saw that you was it yesterday, sequenced your first SARS-CoV-2 genome. So that's absolutely fantastic.
Absolutely.
Anyway, to return to malaria, obviously, it was a great network that you set up, and it's gone from strength to strength. And the science paper that you presented was absolutely beautiful. The things that were really striking to me about that was the kind of the genetic diversity that's structured geographically. And that is something that we've seen. I've worked in microbial genomics for a few years, and that's something that we see with bacteria. And also being able to look at drug resistance. So the great thing about sequencing genomes from bacteria, viruses, parasites is that you can look both at population genetic structure, but also at resistance, antimicrobial or anti-malaria resistance, and also virulence markers. So that was some very, very beautiful work. One question I had about that was, where was the sequencing done for that project? Was it done, for example, at a big sequencing center like the Sanger, or was it done individually by the laboratories?
Oh yeah, that was all done at Sanger, because those days none of us had the capacity to sequence any parasite. We were just doing plain PCR in our respective laboratories, and so all these samples were shipped to Sanger and were sequenced by the Sanger sequencing facility. The analysis, though, it took some time actually to get the sequences done. By the time the sequences were ready, we applied for a grant, which was a bioinformatics development capacity grant, funded by the Wellcome and DFID and other partners, the Deltas initiative. And through that funding, we had started to develop bioinformatics analytical capacity. So the analysis was done between fellows of the Deltas, the Delgem program, with technical support from Dominic's team at Oxford. And still, we would have to travel to Oxford, to Sanger, to work with Dominic's team to be able to do part of the analysis that were not possible to do on the continent. So those days, most of the sequencing was, all of the sequences were done there, and a good chunk of the analysis were done with help from Dominic's group.
Okay. And so in terms of kind of translating what you found, the genomics, into clinical practice, obviously it's alarming to see drug resistance mutations arising, particularly in certain countries. But if the samples were collected some time ago, is this translating into treatment failures, or, what's the time lag between having clinical data and genomic data?
Yeah, I mean, that's a very important question. Like I was saying, the developing bioinformatics capacity has been very instrumental. Actually, we have one of our interns in the Delgem program that contributed enough into the analysis of this work to be a co-author on the paper. So the analysis the capacity is really ramping up on the continent. We are at a stage where the technology and the human capacity is really coming along very nicely, and that puts us in a position where we can be proactive with regard to drug resistance instead of being reactive. As you probably know, with chloroquine resistance, with SP resistance, even with mefloquine resistance, it was too late. By the time we knew that the resistance was there and how it came about, it was too late, because people had already started to die in clinics. So we're learning from that. Now the technology is advanced. We can really look at the genetics and look at early warnings and try to do something about it before it actually appears in the clinic. The lag time really depends on the complexity of the disease, the transmission level. So we at this time, this signature of selection have not translated into bona fide resistance yet. And we are still at the stage where we have to nail down from these loci what are the specific genes that might be involved in this emerging reaction. In the meantime, the known markers of artemisinin resistance, the K13 mutation, have been identified using genetic work again in Rwanda. And the genetic study did demonstrate that it was an homegrown parasite, not an importation from Asia. Yet again, there are still no clinically patent drug resistance yet. So now the idea is to zoom in and eliminate those parasites before they even get a chance to become fully and clinically patently resistant. That's the idea behind the prospect that we can now go in and deal with the emerging resistance, even not giving it enough time to start killing people.
And then I had one last question. We might go a little bit over time, but maybe they can cut it or something.
Yeah, exactly. Yeah.
So obviously you're now building capacity across the continent to do sequencing locally and bioinformatic analysis locally. So you're generating all this beautiful information, but obviously this sort of information is only helpful if you can use it to influence clinical management or policy. So how are you going to do this? How do you engage your national malaria control programs? How do you get them to use the data when it comes to either policy making or treatment guidelines?
Yeah, and that's exactly where we need, in addition to doing the science, we need to start early, engaging with the policymakers, the National Malaria Control Program, ministries of health, so that they understand the concept behind this genetic work, which is fairly new, especially in Africa, and they get to actually appreciate the value of this new tool. And actually, in discussing with them, you sort of not only you can get them to better understand the concept, but they come up with questions that are of interest to them that you can use the tools to generate. So at the end of the day, we think that by engaging with the policymakers, we can reach common ground where they will see the value and understand the results and be ready to take it up. And this engagement has to be done at the national level, at the regional level, and at international WHO level. And we are working on all those fronts.
Brilliant. Well, thank you very much for a really, really interesting talk and also for a fascinating discussion. I wish we could keep talking for the next few hours.
Okay, thank you very much.
Thank you again, Djimde, for your talk, and Estee for joining the discussion. Just a reminder, this is one of 3 spotlight videos focusing on the individual work of the speakers before they all come together on February the 9th for the live panel discussion. Please check the conference meeting website for the full details of the session. Thank you again to our speakers and to you, the audience, and we'll see you in February.